Oh, hello there. I didn't hear you come in. I'm Drosophila, Dr. Drosophila, and welcome to Trinity College. Here at the Smurfitt Institute, we study human genetics. We all know that genes determine the characteristics of living things. They tell organisms what to do and how to do it. But what happens when this goes wrong? When a gene sequence is mutated, an organism cannot read its instructions properly, and this leads to specific genetic diseases. And that is what I'm here to talk to you about. But first, what's a fruit fly like me even doing here?
Genetic model organism is one where you can manipulate genes. You can make transgenic animals. You can make mutations in genes. You can accumulate from a field which has worked on an organism for a long time mutations in every single gene you're interested in. A good genetic model organism also breeds very efficiently. That is, you can get next generations rapidly in your laboratory, and they're inexpensive to maintain. And a really good genetic model organism also should be conserved, should have processes that are similar to the actual organism you are interested in. So you can study in a different organism that performs a function very well. You can study how a basic process works. The idea being, because of evolution, the mechanisms that operate in a vertebrate like a newt would be similar to the mechanisms that would operate in a human. So we work on flies, which also do many things that humans do. Flies can learn to avoid an odor that's associated with a shock. They can find odors quite interesting if they are rewarded in the presence of the odor. If you show them things that are initially interesting, but then nothing of value comes out of it, they get bored. A process called habituation, which happens to us also. And the flies have so few neurons that you can study the process very efficiently in the fly, and keeping a large number of flies costs a very fraction of what happens in a mouse.
I myself have been studying some humans.
We have two versions of each gene. Now, if a disease is dominant, a single one of those genes, obviously these are alleles, so one allele has gone wrong, it's become mutated, and that allele manifests a dominant effect. So every time it passes through to your children, or when you inherit that single gene from a parent, you get the disease. But with recessive disease, two alleles have gone wrong and they've become non-functional as opposed to dominant. So in dominant disease, an allele becomes mutated and it has a dominant effect. So that on its own will dominate the normal gene when it's inherited. But with recessive disease, the gene has simply lost function. So most of the time when you pass on a recessive mutation, we all have recessive mutations, your children will be fine because they have a normal gene on the other homologous chromosome to counterbalance that, to compensate for that. It's only when you inherit two abnormal genes from two carrier parents, you'll get the condition.
This is the allele for brown eyes, signified by a big B. This is the allele for blue eyes, signified by a small b. The allele for brown eyes is dominant. Family trees, also known as pedigree charts, are often used to illustrate the inheritance of genetic traits in families. If a person has one big B and one small b, they are said to be heterozygous. If a person has two big Bs, they are said to be homozygous dominant. In both of these cases, the individual will have brown eyes. If someone has two small b's, on the other hand, this is known as homozygous recessive, and the individual will have blue eyes. This family tree shows how a trait may be passed on from generation to generation. A recessive genetic disease is passed on in the exact same way as this, except the brown allele would instead be the normal allele, and the blue allele would be the mutated allele, which leads to the genetic disease. People with two copies of the mutated gene will have the disease.
And we have two fundamental different types of photoreceptor. The rods give us night vision, which is very important, but the cones give us daytime vision. So what happens with RP? Generally, RP is a progressive disease in terms of the symptoms that develop. So gradually, if you inherit the gene, you will develop night vision problems as the rod cells, the rod photoreceptors, die off. But that's not all that happens. If that was all that happens, it wouldn't be too bad because you can get by quite well without night vision. But unfortunately, as the rods die off, the rods have a supporting role to protect the cones. And so gradually, you develop loss of cones as well. So as the cone cells die, you begin to experience defects in your daytime vision. And what usually happens is your visual fields, in other words, the extent of your vision starts to close in. And that gradually gets worse and worse as the cone cells die off, until you're left with a tiny bit of pinpoint vision, tunnel vision, in the centre of your eye. But unfortunately, even that goes in many, many people, and many people become totally blind.
But what can we do to solve this problem? I know just who to ask.
For many of those diseases, we are going to have to find individual gene therapies. So if there's a disease that has 100 genes involved in it, we may need 100 gene therapies. So that's sort of a specific gene therapy for a specific disorder. And that's what we're finding out, whether it's an eye disease we're talking about, or skin diseases, or cancers, that there's a variety of different genes that actually can cause similar symptoms, and then the disease is clinically classified in the same category. But when you actually look at the molecular level, the disease has a different cause, a different genetic cause, and therefore may need a different genetic solution. So some therapies, or themes of therapies, are being designed based on massaging the problem, like trying to keep the cells alive longer. And when you go for that approach allows you sometimes not necessarily just target the therapy to a very specific group of patients with a very specific underlying genetic problem, but you might have a more common therapy which will be relevant to a variety of patients with mutations in different genes. So one of the things that we're doing in Trinity is using a technology called RNA interference technology, or RNAi technology. This technology is a bit like molecular space invaders. What you can do is you can use very small little molecules to silence or switch off a particular gene. So out of the 3 billion bases of genetic code, you can use about 20 bases of code to silence a specific gene. So we're designing viruses that express those little molecules, that make lots and lots of copies of those little molecules that will silence the rhodopsin gene, which causes dominant forms of retinitis pigmentosa. We're looking at using those little viruses to put the correct gene in the virus and then deliver that directly to the target cell, to the photoreceptor cells. And the delivery would occur by injection into the eye. So that's kind of a funny thought, but you actually can inject under the retina, subretinally, under the retina of the eye, and the virus then infects the photoreceptor cells with the correct gene and makes the correct encoded protein.
Very, very exciting time for people in this area, because we all feel that through our knowledge, our understanding of the genetic basis of these conditions, we're now getting a handle on prevention.
And because we're understanding from the human genome sequence and from a lot of information globally what the causes of genetic diseases are, we're in a very powerful position now to really design therapies targeting what that cause is, which is pretty amazing and powerful stage to be at.